Good morning everyone, I am Lindy Lumayo and today we will be tackled about the variation in genome structure or the numerical changes of the chromosomes euploidy. The organisms are usually diploid. They possess two sets of chromosomes. Variation in the normal diploid chromosome number is termed ploidy. So there are two types of numerical aberrations. So anueploidy has the following two types, the hyperploidy and the hypoploidy. So in hyperploidy, it has also two types, the trisomy and the tetrasomy. Sa hypoploidy, it has monosomy and nalisomy. So in euploidy, it has three types, the monoploidy, diploidy, and polyploidy. So euploidy is a normally organism possess two sets of chromosomes and they are diploid, which is normal for the 2n set. While the anueploidy, it involves addition or deletion of one or few chromosomes to the usual diploid set chromosomes. The aneuploids arise due to the failure of the separation of homologous chromosomes of particular pair during meiosis. So first, let's tackle about the euploidy. So there are two types of euploidy. So the two types is the monoploidy and the polyploidy. So monoploidy is out of two sets of chromosome of a normal organism when one set is lost. The resulting offspring have just one set of chromosomes. So, a monoploid individual has only one set of chromosomes instead of the usual sets. Some male wasps, ants, and bees, for examples, are monoploid because they develop from unfertilized eggs. So, cells of a monoploid individuals are very useful for producing mutants because there is only one dose of each of the genes. Thus, mutants can be isolated. So next, we have the polyploidy. It is an organism having more than two normal sets of chromosomes are called polyploids. So the three sets are called aeroploids, four sets of chromosomes are tetraploids, and five sets and six sets are pentaploids and hexaploids respectively. Polyploidy is generally found among the plants but rarely found among animals. So about 1/3 of all grasses are polyploids. So polyploidy originates in several ways. Polyploidy is the chromosomal constitution of a cell or an organism that has three or more sets of chromosomes. Almost all animals and plants probably have some polyploidy tissues. For example, the endosperm of plant is triploid. The liver of mammals and perhaps other vertebrates is polyploidy. Plants that are completely polyploidy include wheat, which is hexaploid, and the strawberries, which is the octaploid or having an 8 sets. Some animal species, such as the North American sucker or a freshwater fish, salmon, and some salamanders are polyploid. So there are two major kinds of polyploids, the autoploid and the allopolyploid. So first we have the autopolyploid. Are those polyploids which have same basic set of chromosomes multiplied. For instance, if a diploid species has two similar sets of chromosomes, an autotriploid will have three similar sets, and an autotetraploid will have four such sets. So autoploid, it is a type of euploidy wherein the additional set of chromosome is derived from a parent or identical parental species. So in autopolyploidy, there is an additional set of chromosomes which may be form a parent or identical parental species or a single taxon. The cell or organisms in an autopolyploid condition is called an autopolyploid. Autopolyploidy is occur when an individual has more than 2 sets of chromosomes, or the like 4 sets, and all derived from an original species. So autopolyploidy means having many chromosomes of theirs. So during normal meiosis, the chromosomes number is reduced by half. In autopolyploid, a failure in meiosis causes the chromosomes to fail to separate, resulting in gametes with twice as many chromosomes as normal. If these organisms fertilize itself, then its offspring will be tetraploid, having 4 sets of chromosomes. So this is the examples of autopolyploid: the Musa barbiciana, which is diploid, and the Musa acuminata, which is triploid. It contains less seed. Next, we have the allopolyploidy. The set of chromosomes involved come from different, though usually related species. It's much more common in nature than autopolyploidy. About 80% of all land plants may be allopolyploids. So this situation can arise if two different species interbreed to produce an organism with one haploid set of each parent's chromosome, and then both chromosome sets are doubled. So it has many chromosomes from others. Almost all alloploids are infertile due to not having full sets of chromosomes. Full sets of chromosomes are needed for the pairing of chromosomes during the process of meiosis. There is one huge requirement that has to be met in order to form allopolyploidy to occur in its own. The organism must be sexually compatible. This essentially means that it must be possible to get the male and female gametes together through whatever reproductive materials or mechanism is used by both species. So these are the examples of allopolyploids. It has a Raphanus brassica and the allohexaploid web. So let's proceed to the physical characteristics of polyploids. Polyploids tend to be larger than their ploid counterparts. They have larger cells with increased volume. Polyploids tend to display semi-sterility or sterility. Polyploid plants tend to display heightened susceptibility to frost damage and higher resistance to the pest and pathogens. Polyploid plants tend to grow slower and flower later. So polyploid plants have generally large flowers, seeds, and fruits. Polyploidy act as a conservative process and stabilizes interspecific hybrids. Polyploidies facilitate gene exchange between distantly related species. So the segregation and linkage in polyploids. Gene linkage. The linkage of gene on a chromosome results in an exception to the Mendel's law of independent assortment, because linked genes usually do not segregate independently. Polyploidy may also result from doubling of chromosome number in a F1 hybrid, which is derived from two distinctly different species, or the allopolyploidy. The common examples of the autoploidy is the common dovegrass or the cynodon, and those of allopolyploidy is the Raphanobrassica and the common web. Now let's proceed to anueploidy. Anueploidy refers to the presence of abnormal number of chromosomes in the cells. So if we talk about humans, we know that humans have 46 chromosome number in the cell. So this is the normal number, either the presence of higher number like 47, 48, 49, or lower 45, 44, and 43 are the abnormal number of chromosomes in the cell. So if the 46 chromosomes in the cell, then the higher or the lower number of chromosomes are anueploidy. So anueploidy, it involves addition or deletion of one or few chromosomes to the usual diploid set of chromosomes. The anueploids arise due to failure of the separation of homologous chromosomes of particular pair during meiosis. It is known as non-disjunction. So the anueploidy occurs during the cell division. It is when there is any error in cell division with which some cells get more chromosome and some gets less than the normal. When we see during cell division, most of the errors that contributes to aneuploidy are non-disjunction. Non-disjunction of chromosome means disorders of chromosome number are caused by non-disjunction, which occurs when pairs of homologous chromosomes or sister chromatids fail to separate during meiosis 1 or 2 or during the mitosis. So, the diagram below shows how non-disjunction can take place during meiosis 1 if homologous chromosomes don't separate, and how this can lead to the production of aneuploid gametes. Non-disjunction can also happen in meiosis 2 with sister chromatids instead of homologous chromosomes failing to separate. Again, some gametes contain extra or missing chromosomes. Now, let's proceed to what are the types of aneuploidy. So, first is the hypoploidy, which means when the chromosome decreases from the normal. The hyperploidy is when the chromosome increases from the normal. So the different conditions in aneuploidy are monosomy, nalisomy, and trisomy and tetrasomics. So monosomy is the loss of single chromosomes. Individuals are called monosomic and their chromosomal composition is 2n-1. The nalisomy is the loss of both pairs of homologous chromosome. Individuals are called nalisomic and their chromosomal composition is 2n-2. The trisomy is the gain of an extra copy of chromosome. Individuals are called trisomic and their chromosomal composition is 2n+1. And the last is the tetrasomics, the gain of an extra pairs of homologous chromosome. Individuals are called tetrasomics and their chromosomal composition is 2n+2. Now, first of all, let's see what are the different monosomic condition occurring in humans. So, in monosomy aneuploidy, the cell has got one chromosomes number than the normal one. Like, we have the Turner syndrome. The Turner syndrome having X0 condition, also known as having 45X0 condition. This monosomic condition is seen in female, which female is missing the 1X chromosomes. So here in this case, the female has got only 1X chromosomes instead of 2X, that is 45X0 condition. Other monosomic condition is the cridatic syndrome. It is a deletion of chromosome that is called pyaphomic chromosomes. Now looking at nalosomic condition. Here in this condition, the gamete cell do not receive the homologous pre-up chromosome at all. So 2 chromosomes are missing from the cell, which means no complete geometry formation in the cell. Genetic disorders caused by aneuploidy. So this is the figure, which is karyotype of the chromosomes from a person with Down syndrome, showing the characteristics of 3 copies of chromosome 21. So the most common trisomy among embryos that survive to birth is the Down syndrome or the trisomy 21. people with these inherited disorders have short stature and digits, facial distinctions including a broad skull and large tongue, and developmental delays. So normal chromosome complement. So as said earlier, diploid has it is a normal chromosome set, which is 2n, and you can see that this is the figure showing the normal chromosome complement, and the anueploidy, which is nalasomic 2n minus 2, monosomic has 2n minus 1, doubly monosomic has 2n minus 1 and minus 1. Trisomic has 2n plus 1 Cartesian, tetrasomic has 2n plus 2 Cartesian, and doubly tetrasomic 2n plus 2 Cartesian. And now you can see the difference between the types of anueploidy. So now you can see in this figure the difference between euploid and aneuploid. Euploid is the normal chromosome having a 456 chromosome number, which has 2 sets, while in the monosomy or the aneuploid, it has a missing chromosomes, 1 missing chromosomes, and has 45 number of chromosomes. In the trisomy, it has extra chromosomes and 47 chromosome number. So that is the end of my topic. Thank you.